for coming back for our next set of speakers. I'd like to just give a round of applause to our first set of speakers in the morning. I thought they were just marvelous. Thank you. And I think they've set the stage for our next set of really exceptional and exciting talks that you'll be hearing now. And we're going to begin with Dr. Regina Sullivan, and I think Dr. Sullivan will actually be an excellent bridge from the research that we heard in the morning, where we have a lot of animal models that we were working with. And now in the afternoon, we're going to be moving towards more human-based research and questions that are more applied. Dr. Sullivan is professor in the Department of Child and Adolescent Psychiatry at the NYU School of Medicine. And her talk today is entitled Maternal Control of the Infant Brain, Lessons from an Animal Model. Dr. Sullivan.
Thank you, Tracy. So, hope everybody feels better after lunch, and we'll try to give a talk that will bridge this morning and this afternoon. So I'm going to talk entirely about rodent research, but within the framework of human research. So as we begin to think about the brain and development and when we explore the brain to think about how it informs what we heard about this morning, it's important to realize that the infant brain is not an immature version of the adult brain. So, for example, sorry. Okay. Yeah, I got it, thanks. Sorry, these buttons are close to the... So as we think about the child's brain at this age, we don't think of it as being less good at doing the behaviors expressed by the adult brain. So the adult brain goes out and forages, reproduces, etc. The young brain has to focus on the attachment figure, how to be a good infant, how to be a good child, and how to learn how to develop skills that will make them a good adult. So in the young brain, there's two factors that are important to consider. One is not all of the brain functions of the adult are in the infant or the child. First, because some of those brain areas haven't developed yet. But secondly, for the brain areas that have developed, they sometimes have a different function. And what I'm going to show you today really shows how the brain of the infant functions differently from the adult. And when we think about the roots of mental disorders discussed this morning, it's important to consider how experience of poverty and trauma can impact the developing brain, not just because we've disrupted the building of the brain, but because we're implanting experience-based input for brain development. So I'm sure most of you understand the literature on if a child grows up in a stressful environment, then you want to program the brain for a later life stressful event. And working with programming is great. It enables humans to live in many different environments, but I'm also going to tell you that it also leaves the brain open to vulnerability. So early childhood experiences can go beyond programming adaptive behaviors to programming later life psychiatric disorders. So there's a lot of myths in our society concerning mother-infant interactions. A baby will attach to anybody. The baby is politically correct with gender-neutral assignment for the caregiver. A baby will respond to men, women, any sexual orientation, they don't care. And I think there's a big bias in our society that the woman is somehow has a maternal instinct, and it's those maternal instincts that help us able to raise a child. The fact is, maternal behavior is learned. You can get a male rat to respond maternally with all the behaviors except nursing. A male rat can do those behaviors. And when you think about the human brain and human males, they're far more learning-based and So anyway, as we think about attachment and how that influences the brain, we need to understand that it's an incredibly flexible system, and we have so many biases about how it works. And it's our job to get beyond those biases and begin to think about what stimuli from the environment are important in programming the brain or disrupting that development. Okay, so everybody knows attachment makes the baby go to the caregiver. But attachment does something far more relevant to this discussion than just getting the baby to come to the caregiver. So the immature brain in humans and other mammalian species actually can't regulate itself very well. So for example, in children, the brain doesn't have the behavioral mechanisms for thermoregulation yet. So as a caregiver, you must put clothes on and off the child, and you're regulating their temperature. We also regulate the child's, sorry, that's really close. Sorry, I did that. We control what the baby eats and drinks. Maybe I shouldn't use the pointer. My fingers are too big. So anyway, we also regulate the baby's emotions, and we know that we do this. So when we're holding a baby and the baby's a bit fussy, we can get that baby to have a better emotional state for social interactions. And as a neurobiologist, you know that any behavior that occurs must be produced because the brain changed, because the brain is the organ of behavior. So if I've changed that baby's crying into a smile, I've changed their emotions by changing their brains. So it can't be such a far leap to think of how the caregiver regulates the brain. And I'm going to show you some data illustrating how the caregiver actually changes massively how the brain responds to the world. So, and I'm going to do that in an animal model, rodents, rats. And I'm going to justify that by saying that the attachment system is evolutionarily preserved and occurs in many, many, many species, not just mammals, but also in avian species. Any species that requires the caregiver to take care of them, to survive, is going to have some kind of attachment system. We know very little about the attachment system in mammals. I study the attachment, how the infant learns about the caregiver, the neurobiology of attachment. I'm not going to talk about that today because I want to talk to you about how the caregiver can regulate. But anyway, once the infant learns who the caregiver is, the infant has actually learned the sight, the smell, the touch of the caregiver. And that information about the caregiver goes through the baby's sensory systems and into the brain and changes how information is processed. And there is no receptor for love, there is no receptor for attachment figure. It's only sensory information going into the brain. So if I want to understand when something goes wrong with attachment, you can look at how that sensory information is being processed by the brain and how well those sensory stimuli can regulate the physiology of the infant. So I'm going to give you two examples about how the caregiver can regulate the brain. One of them is more similar to a human model. We call that the Scarcity Model of early life adversity. And it's our naturalistic model in which we induce a mother rat to treat her pups roughly. We used to call it our maltreatment model until we realized that that was a loaded concept. We don't know what this rat mother is doing to the baby rat to change their brain. We don't know what maternal deprivation does. You know, in a procedure where the babies get separated from the mother, we don't know. We think that that separation is important, but we have no idea how the sensory input to the brain has changed to make those changes in brain development. So when we have this naturalistic model and we are producing maltreatment by the mother of the baby rat, we have to step back and say, just because we call the model that doesn't mean that's what the mother's actually, that's what's important to the rat pup. So anyway, lots of species maltreat their babies. Along with maltreatment, hurting the infant, usually there's some neglect. as well. There's usually some resources associated with not enough nutrition, so many things typically go wrong as this occurs. So we are calling it our scarcity model to focus on what we're doing differently to this rat. And what we're doing differently is we're giving the mother too few resources for nest building. The mother can't build a nest properly because she doesn't have the material for it. That's the only thing different between our scarcity model, rat mother, and the control animal. When we give the mother too few resources for nest building, she gets a little bit frantic and starts repeatedly building nests. And as she's repeatedly building nests, she's taking the pups back and forth across the cage and ends up hurting them. She also neglects them a bit. So this scarcity model was first developed by Tanya Roth in my lab many years ago. And this just illustrates how the behaviors, the probability of certain behaviors changes dramatically when the mother rat has too few bedding for nest building. We do this for about 5 days from the time the pup is about 8 to 12. We've done it at many different ages, but I'm just going to show you that data today. And I'm going to show you a few different techniques that we've used to understand what this treatment does to the brain. So in this first slide, I'm going to show you that we've recorded local field potentials, LFPs from the amygdala. We've also done it from the cortex. The results are the same regardless of the brain area. Basically, as you can see, you probably all heard about these different names for oscillations in the brain, gamma and beta, for example. What I'm going to show you is that the mother rat, when she comes close to the baby, she suppresses the power of the gamma and the beta waves. So a good mother, when she approaches her baby rats, the brain has less power in that oscillation range. The pup that's been reared with a mother who's been in our scarcity model, that mother can't do that as well. So what happens is the mother has less ability to help the baby rat regulate the brain and behavior. So the ability of a mother to change the oscillations of her rat pup's brain is amazing. During brain development, the level of oscillations and the power of the oscillations in different ranges actually has massive control over which molecules are activated, for example. So molecules that are used to build the brain, for example, are more prevalent at certain oscillations than others. And we know from other research that oscillations are programming, the level of oscillation is programming the brains, such as Carl Lashat's work in the development of the visual system. So this is incredibly important that the mother rat that is maltreating her pups in our scarcity model is actually controlling the pups' oscillations less well. And we're using LFP because this is equivalent in some respect to EEG recording, so it's something that you can do in humans. On a more fine-grained analysis, we can also take a level, we mostly focus on the amygdala. We can take the measurements of how active is the amygdala. So instead of looking at these broad range oscillations in the brain, we can look at how active is the amygdala and how is that controlled by the mother. So the mother controls many, many different brain areas. If a pup is raised by a control mother, you can see that how these bars are, so you can see here's the basal nucleus of the amygdala, the medial, the lateral, and the central. The first bar is always from the control mother, the second one is always from the low bedding of the scarcity model. And you can see that in the scarcity model, in every single amygdala nuclei, the mother is changing that brain area less well. We also know that when you have activity of a brain area, how active it is can also make major changes in that brain area. We know that the mother is changing the dopamine system. In the scarcity model, the dopamine system isn't changed as much by this mother. And just to give you another broad stroke view of what the mother's doing to the brain, is so that we know the amygdala is different, but the amygdala is also talking to other brain areas differently. So if somebody looks at an fMRI of the human brain, as Nim Tottenham will show you in a minute, she's going to talk to you about functional connectivity between different brain areas. And what you can see from here is that a pup reared by a control mother versus a pup reared by the scarcity adversity model of low bedding, the communication between the amygdala and different brain areas is huge. And I think if you think about how oscillations also control how well a brain area talks to one another in terms of long distance and short distance communication, perhaps that's one way in which the connectivity is changed. We don't know. We also try to use techniques used in humans to bridge the human research with the animal research. And here's an fMRI study done in collaboration with Javier Castellanos' lab, in which we show that an animal reared with a low bedding, a scarcity model mother, shows massive differences in resting state functional connectivity for a few different brain areas, including the amygdala and the medial cortex. And also there's work done by Mar Sanchez on non-human primates and Nim Tottenham, who will speak next, which really can use this fMRI research to bridge the non-human primate literature and the human literature to better understand. So I guess the approach in my lab is that we try to mimic what can be done in humans, but then take that information and dig deeper and do things in the rodent that you can't do in other animals. So that gives you a broad scope. And I wanted to show you our broad naturalistic human paradigm because I wanted to talk to you about what kind of information that data can give you. But you know what the problem with that data is that I'm still relying on correlations. And that's what we have to do in humans. We have to rely on correlations. So if I were to take this human literature, this rodent literature, and just do what I can do in humans, I'm not really using the strength of the rodent model. So I'm going to show you our next model. We have probably half a dozen to a dozen different models that we use in our lab. And each model gives us a very, very different aberrant brain development in a very specific phenotype. So we have an anxiety model and a depression-like model, for example. So I'm going to show you the strength of an animal model and how it can help inform humans in a different way that I feel is really helpful. So I'm going to start again with the human issue of maternal regulation of the infant. Remember that anybody can be the caretaker and regulate the infant. I'm using mother because I use the rodent mother. Anyway, so there's this process called social buffering in humans, and in which the mother reduces the stress hormone activation during a painful procedure. So maybe a lot of you know this. If you hold a baby, if you're the caregiver and you hold a baby during the time that they get an injection, that baby actually has a lower pain response and actually has less stress hormone release. It's a massive effect in children. It occurs throughout development. So my husband can probably socially buffer me. And it occurs in many, many, many different species. So this is something really broad. And we've been using this in our rodent model. Anyway, so here's what we do. So we want to make sure that we have validity in our model. And we're going to mimic maternal presence during pain and the reduction of pain and stress hormones. So we have 3 groups of animals. These animals are 12 days old, so they're still halfway to independence. Anyway, some animals just get shocked. Some animals get shocked in the presence of their mother. Their mother's anesthetized, and then some animals get nothing happens to them at all. As we're doing that, we're recording from the brain and behavior. So first of all, just like the human infant, the rodent infant, if you have the mother present during a painful procedure, their behavioral response to the pain recovers very quickly. We're not the first ones to show that. We also show that just like the mother's presence during an injection can reduce crying in the infant, in the rat pup, if the mother's present, they have reduced ultrasonic vocalization. And then finally, just like the human, in the rodent, if the mother's present, there's this reduction or blunting of the stress hormone release from the shock. So if you just shock the animals, you get this big release of stress hormone. If the mother's present, you don't. So again, why would I do that in rodents if we know that in humans? We already know that, so I'm not contributing very much at all if I do that in the rodents. I'm just doing that to see, to show validity of the model. Okay, so here's one thing I can do in a rodent that I can't do in a human. I can look at where in the brain is the mother possibly able to block stress hormone release. We have no idea where that occurs in humans, so this piece of information can tell you something about the mechanism. So we started at the hypothalamus of the HPA axis and a particular brain area called the PVN. So we know that norepinephrine is the major neurotransmitter that activates the HPA axis. We know that from tons of other animal research. So we looked at norepinephrine in the PVN, and we stuck a cannula into the PVN and withdrew the fluid from the brain to measure what neurotransmitters are changed when the animal gets shocked or when the animal gets shocked with the mother present. And here's microdialysis. So after we take that fluid out of the brain, we stick it into a device called the HPLC, and we can measure how neurotransmitters change. So from the top, you can see that here's an animal, norepinephrine is increasing if the animal's just getting shocked. This is a 12-day-old animal, 12 to 14-day-old. This is what norepinephrine looks like if the mother's present during the shock. Complete blocking of the activation of the HPA axis. So the mother is stopping the HPA axis or the stress response at the level of the paraventricular nucleus. But the mother's effect doesn't stop there. We know that pain, even in baby rats, can activate the amygdala, a brain area associated with affective pain and fear. So the middle bar shows a PUPS activation of the basolateral amygdala, a very specific nuclei in the amygdala that's associated with pain and fear. And you can see that there's good activation, solid activation of that brain area. The mother's present, that amygdala activation is completely stopped. So you might think that that's amazing. The mother is actually blunting a brain area that would normally respond to pain. So when we see the behavioral response of the baby, we can think of that being blocked, brain areas associated with the pain actually being blocked. And Nim Tottenham has also shown that in humans. The human mother can block the amygdala activation in children. I think they're 4 years old. And I'm gonna tell you one more thing that happens. Actually, if the mother is present during the shock, she can actually prevent the rat pup from learning about that shock. So think about a child being hurt, and we don't know for sure that this occurs in children, but think about a child or an infant rat being hurt, and then the mother's presence actually blocking the brain area designed to have that baby learn about fear. So the amygdala is the main brain area of the baby rat, adult rat, and us for learning about fear. So when the mother is blocking that shock's pain response by the amygdala, she's also blocking the mechanisms for plasticity to learn that they should be afraid of that, of anything that predicts that pain. So for example, you might swab a child's thigh with alcohol before you give it an injection. The human brain would normally pair those two stimuli together so that when we smell alcohol, we would think of that injection. The baby rat can't do that until it's about 10 days old. So we're talking about a young infant. So I want to explain, because that is so important, I gave you this, I'm going to talk about a cartoon. So here you take a baby rat. If you pair pain with an odor, a baby rat can't learn fear, because the amygdala isn't activated during this learning process. We call this our animal model of abuse because as a rationale for why children who are abused by their caregiver don't learn to avoid their caregiver. If animals are older than 10 days, see, I can't touch this thing. It's like, if you take an animal older than 10 days of age, if you pair an odor with shock, they can learn to fear that odor. And that involves the amygdala. If we block out the amygdala, that animal can't learn to avoid that odor. But the amazing thing that happens at this age, because the mother entirely blocks the amygdala response to the shock, is that we can take this older animal, place it with the mother, and you actually get a more infantile brain mechanism or circuitry occurring. So when we think that this informs us about how the infant brain processes trauma. So if the infant brain is processing the trauma without the mother and with the mother, two different ways, that's incredibly important information that helps us understand that trauma might be different. The long-term effects of trauma when experienced alone or experienced with a caregiver are very likely to produce very different outcome. And I want to also mention that the circuitry in the brain to do this gets turned on and off immediately by maternal presence or a social figure. So I don't want anybody to think I'm the only one in the world studying the effects of social partners on the amygdala. There are many people that do this in terms of social buffering, but also the effects of a social partner being present in a rat cage during fear conditioning, or a social partner being present during learning of fear in the adult. So I told you about how the normal rat pup, reared by a normal mother, has this thing called social buffering, which turns off the amygdala. I'm now going to show you some data that shows if I do that a lot for 5 days, I obliterate the mother's ability to stop the amygdala from being attenuated. So remember, the attachment figure is learned. When that rat pup or that baby is learning about the mother, they're learning the sights and smells of the caregiver. If I take that information from that caregiver and I repeatedly pair it with pain, that baby is continuing to learn. We know from the attachment system that they're still going to attach. What we see is that there's degrading of those stimuli associated with the caregiver. So as these rat pups, I'm going to take rat pups from about 8 days to 12 days old, and I'm going to shock them repeatedly in the mother's presence. And this again was done by Maya Opendeck. So the same procedure I showed you for social buffering, except we do it for 5 days. And then we test, okay, how has this changed how the baby rat responds to their mother? So on 5 days, we pair shock with the mother, or the pup gets shock when it's all alone. And here's some data showing how the maternal response to the mother changes over the course of those 5 days. So here we see that there's pups on the very first day of this treatment have high response to shock if they're alone. If they're with the mother, the response to shock is greatly reduced. By the 3rd day of the 5-day treatment, these rats are starting to respond more to the shock. So the mother's ability to suppress the response to shock is decreasing over time. You know, the red is showing that the baby rat is responding differently to shock alone, and that's a whole nother line of research on pain in the lab. A repeated pain to a baby is not good in many, many ways. What we're suggesting is that you have pain with the caregiver, it produces a very different response in the brain. But also it shows you that the mother here is no longer attenuating the ultrasonic vocalizations anymore either. So the baby is responding to pain a lot more. And this is a test done after the treatment is over. There's no more shock. The babies are absolutely just responding to their mother in a normal situation. If we look at these pups in the nest, we can't tell who was the pups that got shock and who didn't. So within the nest, the mother makes sure that everything is okay. If we take the pup out of the nest and give them a very controlled experimental test where we anesthetize the mother so she can't help them, what we uncover is that the pup's ability to interact with the mother is greatly reduced. So, in fact, pups with the mother are actually having a very difficult time responding to the mother at all without the mother's help. So just an anesthetized mother suggests that their social behavior with the mother is greatly disrupted. The amygdala nuclei are massively changed. So remember I showed you a slide that said the mother can blunt or block the activation of the amygdala if the pups get shocked. 5 days of that treatment, the mother is no longer able to block amygdala activation. I know this slide is a little bit complicated, but I wanted to let you know that for the central amygdala, the response to shock is decreased in pups, and the first day of treatment, the shock with mom pups are up here. After 5 days of treatment, you can tell the difference between a pup getting just shock or shock with the mother. So the ability of the mother to attenuate the amygdala is gone, or to activate the amygdala is gone. So, so what is the big picture information that we get from this information? Well, we know that infant attachment is learned, and that learning system is incredibly robust because it's phylogenetically preserved and occurs in many, many different species. Once the attachment figure is learned, the attachment figure acquires the ability to regulate the infant's behavior and brain and physiology. These attachment sensory cues are important for modeling the pup's brain response to sensory stimuli, including trauma. So remember, if we just do it for 1 day, the mother can attenuate the brain's response to trauma. If the mother is repeatedly paired with trauma, the mother loses her ability to regulate the pup's brain as well. Anyway, that's enough for the conclusions, actually, and I'd like to thank my collaborators. Maybe some of you met Maya Opendek. She was giving a poster at lunchtime, but had to leave. Anyway, thank you so much. Oh, right, I write questions later. Yeah, thank you.
Thank you so much. That was wonderful. And I think we have a great segue now to our next speaker, Dr. Nim Tottenham, who's Associate Professor in the Department of Psychology at Columbia University. And she'll be speaking today about early experiences and cortical subcortical brain development in humans.
Okay, good afternoon. So our laboratory is addressing something that's been a focus of today's session. Ooh, that does go quickly, which is emotional functioning and maturity. And the variable that we're most interested in are the early experiences that we have, both the adverse experiences, but also the positive experiences that we have early in life that contribute to this enduring emotional phenotype. We are very focused on the development of the connections between the amygdala and the medial prefrontal cortex, as well as the hippocampus, in large part because the circuitry forms the foundation of many of the emotion regulation behaviors that we see in adulthood. The amygdala, an earlier developing region, is a site that supports emotional attention and emotional learning. And in maturity, we have these strong bidirectional connections between the amygdala and prefrontal cortex that support emotion regulation abilities. Our approach is to study the system during development. So I liken it to, I'm the type of person when I'm cooking a pie, I keep opening up the oven door to check on the progress of the pie. And I think sometimes you get different answers when you're looking at how the system is responding during development following particular early exposures. So I wanted to share with you briefly some of our findings on typical development of amygdala and connections with the prefrontal cortex across childhood, adolescence, and early adulthood. So in response to simple stimuli like fear faces, if we look at age here on the x-axis and amygdala bold signal on the y-axis, what you can readily appreciate is that the younger you go on this scale, the more robust the amygdala response is to something like fear faces, which makes a lot of sense because when you're young and when you're new to this planet, you have a lot of learning to do about the relative safety and danger of the environment, so you want the amygdala chugging at full steam. However, when we look at amygdala functional connections with the prefrontal cortex, we get a different picture. So in adults here, 18 to 22-year-olds, we get this very prototypical anti-correlation between amygdala and prefrontal cortex, which is really indicative of a regulatory relationship between the two regions. Older and younger adolescents also show this anti-correlation, although you can see that it's not as strong as the negative correlation that's observed in adults, which is consistent with what we know about the slow development of emotion regulation abilities. However, what we were most struck by was what we observed during childhood. So during childhood, we don't get that anti-correlation or regulatory relationship between amygdala and prefrontal cortex. Instead, we get this positive correlation between the two. So the amygdala and prefrontal cortex are working together rather than in opposition in response to fearful face stimuli. So we've since become very interested in this developmental switch in communication between amygdala and prefrontal cortex. So at the very least, what we can say is that it seems that between childhood and adolescence, there's this big change in the way that amygdala and prefrontal cortex are talking to each other in response to emotional stimuli. So putting these two types of data together, if we put age on the x-axis here, what we're seeing is that during childhood, we get a very robust amygdala response, but this is happening in the absence of the adult-like connections with prefrontal cortex, and I can add hippocampus into there as well, which makes childhood a very interesting time for us to think about how environments can shape the way that this system is going to construct itself over time because the phenotype is so fundamentally different. Moreover, it may be that this high amygdala activity, which is normative in development, may be a necessary prerequisite for the instantiation of these regulatory inputs from prefrontal cortex. So I'll share with you our current interpretation of these developmental changes in connectivity. It may be that, given the hierarchical nature of brain development and that the amygdala is an earlier developing region, that the connectivity we're seeing early on is really reflecting the amygdala driving the conversation to the prefrontal cortex. So I often think about the amygdala as an older sister who got there first and learns about the world and starts to instruct the prefrontal cortex about what it's learned, so that over time that communication can help instantiate the reciprocal connections from prefrontal cortex. And there are now many nice tracing studies in rodents, including a recent optogenetics paper that would support this idea, showing that amygdala originating inputs to prefrontal cortex are emerging developmentally earlier than the reciprocal connections. So one of the questions that is really driving a lot of our research is childhood a sensitive period for the development of the connections between medial prefrontal cortex and amygdala? And we've been inspired by some work that Takao Hensch's group has done using music learned during a juvenile period as a probe for sensitive periods. This ended up being very handy for us because studying or trying to identify sensitive periods in humans who take so long to grow up requires either a time travel machine or some stimulus that you feel certain about the age of exposure. And music, at least pop music, may give us some ability to do that. So if we want to know about a childhood exposure, in other words, something that happened at age 7, and we're studying a young adult in the year 2012, then we want a stimulus from 1997. So we can use data from the Billboard charts to know how old our young adults were when they were exposed to particular stimuli. So this is a study in collaboration with Tikau Hench. Laurel Gabbard-Durnham, a former graduate student, has done this very nice study. She brought in undergrads and gave them a lab stressor. It was hard SAT math. These were UCLA students, and she said, you're doing pretty well, but not as well as your classmates, so try and pick up the pace. And then gave them rest bouts. And during these rest bouts, they could listen to one of two radio stations. One would play music from their childhood, so this is Quit Playing Games with My Heart by the Backstreet Boys. And the other radio station would be songs from their late adolescence. So this is Justin Bieber and Ludacris, Baby. And so the question is, under duress, do we see a behavioral proclivity for one of these radio stations? And what Laurel has found is that with increasing stress bouts, participants increasingly choose the childhood stimulus. Sorry. If she breaks this up into people who report having a lot of exposure, that green line jumps up even further. She had a control group of participants, I'm gonna try this, who were not born in the US, reported not listening to American music, and they're not choosing the Backstreet Boys because they weren't exposed to the Backstreet Boys. This is not about liking or hedonics, because nobody's walking in saying that they like the Backstreet Boys, but under duress, they are increasingly showing that behavioral preference. However, it was only those individuals who were exposed to their childhood stimulus who also showed significant decreases in autonomic arousal. The more that people chose their childhood stimulus, the increasing reports of calm. And during scanning, it was the childhood music stimulus which selectively increased this medial prefrontal cortex activation, which is very similar to what was observed in a rodent study. And the more that medial prefrontal cortex increased its activity, the calmer people felt. And I should mention that this medial prefrontal cortex activity was associated with strengthened connectivity with the amygdala. So this was a very fun study to do, but of course we're not interested in what the Backstreet Boys do to your brain. We're interested in species typical exposures that coincide with this putative sensitive period. So the circuitry is not developing independently in a test tube. It's developing, as Regina so nicely described, in a very special species expected context, which is the caregiver. And as Regina said, this is a phenomenon that occurs in many species, but what is important to point out in humans is how long humans stay hanging around their caregivers. So in many species, in most species, we're talking on the order of weeks or months, and in humans, it's on the order of years or decades, depending on your family structure. And it's been argued that one of the benefits of this is that this gives humans an incredibly long period of neural immaturity or neuroplasticity that benefits us in the long run. And so parents can serve as a social scaffold that supports central nervous system development. So we just heard from Regina, but I'm going to briefly describe a set of findings that she didn't highlight, which is to say that there is this period during development and rat pup development where the functioning of the amygdala is dependent on the mother's presence, as she described. Behaviorally, this works out such that when a rat pup is placed into a Y maze, with a fear conditioned stimulus, for example, a peppermint odor on one arm and a control on the other, this rat pup will avoid that arm with a peppermint odor, just like you and I would because their amygdala is engaged. However, if you take the same rat pup and you place its mother next to it, then that mother will block amygdala activation and also block fear learning and promote preference learning in a way that is species adaptive for attachment learning. So Regina also has another very nice paper showing that this effect depends on a regulated mother, right? So this mother is very regulated, she's anesthetized. If the mother herself were expressing fear behaviors, then she would have an equally powerful ability to promote fear learning and amygdala engagement. So in this way, we can think about the parent during development as being a privileged information source to emotion learning systems in the developing animal. So given that human development is so stretched out compared to rodents, we were interested to know whether there would be any evidence of this parental buffering or modulation of amygdala activity. And what we have found is when we present children with pictures of their parent and compare them to pictures of a stranger, we see that during childhood, the parent is reducing amygdala activation compared to somebody else's parent's picture. Note that in adolescence, there's no difference there. Parents aren't modulating. It's not to say that parents aren't important during adolescence. Of course they are. But it may be that at the level of the amygdala, the parent has lost some of their ability to modulate this activation here. So we think that, at least at a phenomenological level, the parent is able to buffer amygdala activation in the human as well. Whether the same exact mechanism is occurring here as what's happening in the rodent, we don't know, but at least at face value, we're getting that buffering effect. And also Megan Gunner's group has shown this at the level of court response to a Trier social stress test as well. So we wanted to know whether this difference in the amygdala activation would correspond to differences in fear learning. And so we really just used, very much modeled this paradigm off of the work that Regina's lab had been doing. And we presented young children with a threat conditioning paradigm where this blue square would be paired with a loud, nails down a chalkboard kind of noise, so it's not painful, but it's aversive. And the purple triangle never would be associated with anything. And children learn this next to an experimenter or sitting next to their regulated parent. Following learning, children were placed into a human Y maze. Children would enter a tent, and this arm would have the blue square and this arm the purple triangle, and we would ask them to go through and pick a door to retrieve a prize, same prize behind both doors, and the DV was which door were they likely to choose. So I have a very brief movie here just to show you if it plays. Yeah, so this little girl is going to enter the tent, and she's going to look at both doors with the CSs on them and make her selection. That's not an injury, that's how we collect galvanic skin response from children's feet. Okay, and she goes in to collect her prize. So these are data from 3, 4, and 5-year-olds. Lower scores here mean that they're avoiding that CS+. If children conditioned sitting next to an experimenter, they did more or less what you and I would do, they would avoid that blue square. But if these same children were conditioned sitting next to their parent, then their aversion learning wasn't so good. And in fact, as we go to younger and younger ages, you start to see some of that seemingly bizarre but developmentally appropriate approach behavior of the blue square that had been paired with the aversive stimulus. So this gives us some ways to start thinking about the development of emotion regulation circuitry in children who do not have access to a parent. So children who grow up, for example, in institutional caregiving, which we often call orphanages, even though they're not technically, experience a species atypical caregiving environment. It's a lesion of the parent stimulus in a way. And there are massive individual differences that I will try and point out in the rest of my talk, but suffice it to say that at the very least, it's a suboptimal caregiving situation because the caregivers are staff members who rotate shifts and the caregiver-to-child ratios are incredibly poor. If we look at the rodent literature that has examined the influence of early caregiving adversities, what many labs have been finding now, and we've heard a little bit about this from Regina, is that early life stress can accelerate the development of emotion regulation neurobiology. And the argument is that these differences are developmental adaptations that the organism makes in order to meet immediate needs. And the question is, if you make an adaptation to meet an immediate need, is there a potential long-term cost that you want to be watching for? So you want to think about the temporal dynamics of these neural phenotypes. So children that have been adopted from institutional care experience a very different early experience compared to a later adoptive home. The families that adopt internationally are well-resourced, but most importantly, have a great desire to provide care because it's not easy to adopt internationally. Now, although many children are at risk for developmental delays upon adoption, there's significant rebound across a number of domains following adoption, perhaps not surprising given what I've described about the families. There's also a significant heterogeneity. Some children are thriving by every index that we can come up with, they're thriving, and some are struggling. If children are struggling, it is very common for parents to express their concerns about emotion dysregulation or behavioral dysregulation. So we have, when I was training with BJ Casey at Cornell Sackler, we worked with families that had adopted children who were previously institutionalized. And then when I moved to UCLA, we collected data from another cohort. In both groups, we observed elevated anxiety, regardless of what subscale we looked at, but most, not surprisingly perhaps, the biggest area of concern was separation anxiety. And in both cohorts, we observed that a history of institutional care was associated with hyper-response, hyperactivity of the amygdala to emotional stimuli. When we look at amygdala connectivity to prefrontal cortex, this is functional connectivity again, we find important caregiving group by age interactions. So these are the data I showed you earlier, where children with a typical caregiving history show a more childlike, non-regulatory relationship between amygdala and prefrontal cortex that switches in adolescence to that more regulatory adult-like connectivity. We were able to replicate that in a separate cohort. But when we look at children in this group with a history of institutional care, we don't see the childlike connectivity pattern, but instead we see the more adult-like regulatory pattern. So this is consistent with the idea of some type of accelerated pattern of connectivity. And if you remember in Regina's talk, she also showed that there was hyperconnectivity not only between amygdala and medial prefrontal cortex, but also hippocampus. And we see that pattern as well with hippocampus. So we're thinking that this difference here is an adaptation. It's a brain difference, but it could potentially be helpful to the child. And I'll explain how we look at that. So what we think is happening is, here's age on the x-axis again. If high amygdala reactivity is a prerequisite for the instantiation of these adult-like connections with prefrontal cortex, then it may be that growing up without stable caregiving, which activates amygdala at an earlier and higher rate, might instantiate these connections at an earlier time point and possibly reduce this putative sensitive period during childhood. But what we see is that this adaptation is associated with current resilience or current lower anxiety. So on average, the history of previous institutional care is associated with elevated anxiety, but if children show that more adult-like brain connectivity pattern, then they show lower anxiety than their peers that did not show that difference. And if I'm getting it right, I think this is similar to Koff's data showing that in the context of behavioral resilience, sometimes you get the most different brain phenotype. So the last set of data that I'll show you is actually performed by my postdoc, Bridget Callaghan, who you'll be hearing from shortly in a flash talk. She wanted to know, okay, so if there is this abbreviated immaturity in some children following early caregiving adversity, are there consequences that we should be looking out for in terms of long-term anxiety phenotypes? So she asked whether this early adversity placed limits on the ability of the parent, the adoptive parent in this case, to buffer against amygdala activity. So these are the data I showed you before, where children with a typical caregiving history show a dampening of amygdala reactivity or a buffering. For a history of post-institutional care was not associated with this buffering phenotype, and if anything, there's a slight potentiation. But what you are probably appreciating is that there are very large error bars on this figure, which means that some children are not showing buffering and some children are. And so we can interrogate those individual differences over a 5-year period to see if this phenotype is predicting longer-term outcome for anxiety phenotypes. And it does. So we find that this parental buffering phenotype at time 1 predicts lower anxiety in the future in all children, but especially so, as you can see here, in children following a history of institutional care. So children who show the buffering of the amygdala at time 1, although there's no difference in anxiety between them and children who didn't show a buffering phenotype, over a 5-year period, they show sharper declines in anxiety phenotypes. And what variables are associated with this difference in showing buffering versus not buffering. This was maybe one of the few times in my career where age of adoption was not predicting this difference. It was not age of adoption, but instead it was the security of attachment with the caregiver. So children who showed that amygdala buffering at time 1, even though they had very high anxiety, they were children who had higher attachment security than their non-buffered peers. So to conclude, human amygdala medial prefrontal circuitry, and I would add hippocampus, develops very slowly, perhaps by design, and that there's great value during childhood or the juvenile period for learning about the environment in ways that can possibly be leveraged in the future. Early caregiving experiences shape the neurobiology of emotion regulation with parents as social regulators during immaturity, and perhaps parents are even helping to maintain this long plastic phase during development. And early caregiving adversity may change development by changing the pacing of development through activity-dependent processes with short-term benefits, but longer-term costs as a risk that we want to be mindful of, even in children who are doing well initially. And so we think that understanding brain phenotypes following exposure to early adversities are best understood through the lens of developmental adaptations. So with that, I will acknowledge the lab members who have contributed to this work, and Bridget should be bolded there as well, the funding sources, and also the families and individuals who participated, and thank you for your attention.
Hi, everyone. So as you'll see from the schedule, we have a brief coffee break now. To keep on schedule, we will just be taking about 10 minutes. Please come back at 3:45. We'll start with our flash talks, our data blitz, and our 2 remaining speakers and panel discussion. Thank you. See you shortly.